Basically what we're going to walk you guys through is an experience about biology and how biology reacts with everything that we're doing and the subtle forces of growing plants.
Can you guys hear him?
Is that a little better? Okay. I'm going to go through this pretty quickly because I guess we've got an hour and it's 25 minutes for myself and 25 minutes for Ian and then a little bit of time for questions. So if there's any questions, just hold them to the end and we'll answer them at the end.
If you run over your time, don't worry.
Okay.
Finish your thing.
Cool. So our goal today is to motivate you with an essential understanding of the practices and techniques that will help you to deliver the highest quality medicine and the most nutritious foods while preserving a healthy growing environment season after season. The topics we're covering are working with biology, the soil-to-plant exchange, the complexity of cycling of nutrients, the soluble and insoluble elements, soil testing, understanding protein synthesis and proteolysis, the functions of soil microbes, photosynthetic capacity, the levels of plant health formation, all the things that it takes place in order to get the metabolites that we're looking for, formations of complete proteins, critical points of influence and epigenetics, basics of plant physiology, regular application guidelines. So here we go.
That's a lot of stuff.
It is. I don't think I'm going to cover this much. So one of the main things when you're working in any type of growing situation, it's a concept of mutualism where you're working with biology instead of against it. Microbes and other organisms, they actually ingest. That's as loud as, is that better? Okay.
Turn it up.
Is that better? Okay. So microbes and plants have a unique relationship where whatever it is that we're growing has a cyclical cycle where it cycles back into our nutrients for the following year or for years to come. And so the nutrient exchange that's taken place with the fungi and the nematodes and bacteria and the plant minerals that we're adding, this is all basically, again, around high biological agriculture, not around chemical fertilizers. Anytime you're using chemical fertilizers, you basically take away mutualism and you start creating kind of an elaborated hydroponic system out in your field. So whether you're doing indoor or outdoor, the main thing that we're doing is focusing on building biology to start creating stable humic substances and to have good colonizations for the bacteria to actually do what we're hoping the chemical fertilizers would do. The soil-to-plant exchange, this is just giving kind of a breakdown of how the plant is basically reusing plant residues and root exudates from not only just that growing of that specific plant, but from prior things that you were growing. So if you're growing cover crops and so forth, there's all kinds of different things that happen with components that are created, the slow decay, the humus, and then how it goes back into a rapid remineralization and a slow mineralization. And the main reason of showing you this is to kind of give you an idea of the complexity that needs to be in place. It's very similar to the gut or the flora of your body. You take antibiotics, it's all gone, you've got to restore it. Use chemical fertilizers, all the flora is gone in your plants and you have to restore it. This is what takes place in the concept of a nitrogen just in itself and the cycling of it. There's so many different processes that it takes in order for the microbes and for the bacteria and the enzymes to actually get the nitrogen into a usable form for your plants. It's not just a simple process of adding something that's very high in N. So if something says that it's 10% N, 20% N, 30% N, whatever it is, typically your plants are only going to use minimum 2%. Maximum that a plant can use from chemical fertilizers is around 12%. So these high numbers that are on a lot of these products, your plants can't actually even use them. These are all the essential elements, and just to kind of give you an idea of the coevolutionary aspect of these minerals inside your soil. Boron activates silicon, which carries all of the other nutrients, starting with calcium, then binds with nitrogen to form amino acids. The DNA and the cell division of the amino acids form proteins, and tag trace minerals, especially magnesium, to form chlorophyll, which transfers energy via phosphorus to carbon to form sugars, which go to where, potassium carries them through. So this is just kind of giving you an elemental concept of all the elements that are in the elemental chart that are necessary for a plant to grow very healthfully. This is a breakdown for soil testing. There's different types of soil tests where they can do what's called a mining assay, or they do different types of tests that will actually determine whether you have a strong acid, weak acid, or no acid test, so they can actually tell what the extractables are that are in the soil. So most of the people that are growing usually buy a lot of peat moss or preconditioned sterilized soil, and they add maybe some earthworm castings or some rock dust or something to it. But it's very rare that you're actually seeing anybody these days where they go out in a field, till up an area, plant a cover crop, and come back and actually plant in that area. Soil testing doesn't really correspond to soilless medias. It's kind of, that's more of just dealing with pHs. Why do we care about what soluble nutrients are? There are forms that the plants pull into the roots through the process of simple diffusion. Basically, the organisms in the soil can make them plant available if the whole food web is working correctly. And one of the things besides just the bacteria is the overall conductivity, and conductivity corresponds to the electrical energy flow in soil. Soil energy levels need to be sufficient for crops to have access to the nutrition needed for optimal growth. Biological activity releases the minerals into soil solution, which increases your conductive reading. So I don't know how many people work with an EC reader that actually look at the EC of your soil, but you can actually see when your biology is, when it's getting at its prime. It's similar to using something that's a refractometer, which I'll go into, but there's some basic tools that you can use, an EC reader and a refractometer, which can guide you to know that there's optimal nutrient flow in your plants.
Christopher, what do you mean when you say the biology in the soil?
The biology in soil.
What's the biology?
Everything. Why don't you break into that after? Because I'll go through all these.
Just give them a, like, what do you mean by that? It might be a little confusing.
How many people in here know about biology in soil? Okay. So basically, your nematodes, your mycorrhizal, all your bacteria, all of the different life forms that are in the rhizosphere and beyond the rhizosphere. One strand of a triticale root, which is like a rye plant, can be over 2 miles long after that plant's in full growth. So there's all kinds of different anaerobic and aerobic bacteria and life forms that are in different substratas of the soil. You want to share anything?
Just, we're talking bacteria, fungi, protozoa, nematodes, all these different organisms that together can kind of be referred to as the biology in the soil. So,
let's see here. There's a lot to cover. I actually packed what I'm pulled is slides out of a 7-day course that I teach with real farmers that are out in the field growing alfalfa and strawberries and onions. It's a process of time. This is a concept of what you're looking at as far as a saturated paste test. This would be if you were sending your product off, your soil, to a laboratory to get a reading back. This is actually an electron microscope of calcium carbonate from your soil. I just put that in there just to kind of give you an idea that these things all have a pretty amazing fractal holographic energy field to them. So they're not just some static thing that you're reading, magnesium and potassium. So what functions do the soil microbes perform? They maintain aerobic soil conditions in the soil. They build soil structure, disease suppression mechanisms, decomposition, nutrient immobilization, nutrient mineralization, and nutrient fixation.
So that's like basically everything that a plant needs in order to grow. These organisms can provide, right?
Exactly.
There you go.
You want to explain this one?
Sure. This is the soil food web diagram, and what it really explains is how plants are fertilized in a biological system. So if you look at the chart, it kind of begins over there with a living plant, and what I'm going to explain here is how microbes can fertilize plants in nature, right? So you have this living plant that dies. So now we've got dead stuff, right? The above ground part of the plant and the root system. And say in the tomato plant in the fall time falls over and it's dead. So now you have that dead material and there's all these nutrients that are held within it. So the blades of the grass, the roots of the grass, whatever the plant is we're looking at, there's all this nitrogen and phosphorus and sulfur and all this good stuff that's tied up in that plant, right? So when it dies, the fungi and the bacteria come in and they eat the plant. So like if you think of the nitrogen that was in the leaves of that grass, it's now tied up in the biology. So you have a bacteria that comes in, chomps away at the leaf, and now that nitrogen has moved into the bacteria. So the next level, the nematodes and the protozoa, they're like predators in the soil. So they'll come in and eat away at that bacteria that ate the leaf, right? So the nitrogen moved from the leaf into the bacteria, and now you got this protozoa that comes in, eats that bacteria, and poops out the nitrogen in a soluble form of NH4 that the plants can uptake. So, you know, in a healthy soil, you might have 1 billion bacteria per gram, and you might have 100,000 of these little protozoa. And each one of those are eating 10,000 bacteria a day, pooping out all that nitrogen. So if you have all these organisms functioning in your soil, the plant can really be fertilized for the most part just by the activity of these organisms eating each other. So it's kind of how the soil food web works. It's a predator-prey interaction that's going on there. So you can think of the predators, nematodes and the protozoa, as the fertilizer spreaders, and the primary consumers, the bacteria and fungi, as the bags of fertilizer. So the more biology you get in your soil, the more of this fertilizing is happening naturally, and you don't need to add so much fertilizer to get the high yields.
That's correct. So the next thing is about turning sunlight into sugar and just the processes of there's so much that goes into plants and how they grow. So basically to bonding together of CO2 with H2O to make a sugar and oxygen using the sun's energy. Successful photosynthesis is possible when there are minerals available to act as a catalyst, adequate air and radiant energy. I don't know how much I'm supposed to be going. A lot of this, when I shared this with the group here at Deep Green, they said a lot of this is going to go over people's heads. So just sounds good.
Maybe learn by immersion here.
Yeah. So basically, the photosynthesis produces complex sugars, and those complex sugars are what builds up the aspects of the proteins and the plant's secondary metabolites, which is what typically, whether it's lycopene in tomatoes or the actives in chrysanthemums or the plant secondary metabolites of THC or CBDs, any of those types of things are the concept or the wording of plant secondary metabolites. This is basically about the photosynthetic capacity. Leaf density, plants are highly loaded with leaves, have a higher productive capacity. Leaf sap color, if people are getting into looking at the sap color, you can tell that that's where the bricks and the refractometer will help you to be able to tell of the nutrient uptake of your plants. Leaf thickness, the thicker is better. The mesothelial cells that are inside the center of what you call a leaf is actually chlorophyll filling out little chloroplast cells, and that's where all of your minerals. The higher your bricks reading, which is actually the reading that's inside of the leaves, will actually tell you how much nutrient flow is getting into the leaves themselves. So this is a good demonstration of how your plants, basically, it's forming sugars and amino acids. It takes water with CO2 to create glucose, which ultimately allows for photosynthesis. That glucose plus nitrogen then creates what's called amino acids. Then you go into the next level of plant health of forming peptides and proteins. And that's where amino acids then, with enzymes, which are basically cofactors which split these amino acids and divide them into peptides. Those peptides then work with enzymes to create complete proteins in the plants. This is the levels of plant health. The main thing that you're working on is you're starting down at the bottom, where when you have successful photosynthesis, you've got the formation of complete carbohydrates, such as pectins and other polysaccharides, which build resistance to soil-borne fungal pathogens, such as Fusarium, Alternaria, and Verticulum. Level 2 is basically when you get the production of complete proteins, the transfer of sugars to the roots, to soil microbes, that then release nutrients in a plant-available form. Increased resistance to insects with simple digestive systems. Level 2 is when you get a resistance to aphids and whiteflies and larval insects such as cabot earworm, alfalfa weevil, tomato hornworm, and many others. The storage of supply is your next level of plant health, and that's where the energy is stored in a form of lipids. So the plant now has created a solid form of carbohydrates. It's built a series of complete proteins, so it's stacking in function. It can start to store and create surplus energy in the plant. When a plant's highly stressed, you'll actually see that it actually depletes its own proteins to keep itself in a resistant form. So if it goes through a drought, if it's got major insects, if it has downy mildew, whatever those types of things are, the plant actually starts to pull into its own reserves, which lessens the overall output of what that plant's able to give you. Lipids build strong cell membranes for increased resistance to airborne pathogens and parasites. And then finally, you get into the production of plant secondary metabolites, PSMs. They act as a plant protectants to guard and against ultraviolet radiation, disease, and insect attack. And if you want to chime in at all, always do.
Go for it.
This is just to give you, this came out of the British Journal of Pharmacology. This is some of the plant secondary metabolites that they're focusing on in the cannabis plant. Just to kind of give you an idea of how many different things have to stack and function in order to create this surplus of PSMs. I mean, there's an endless amount of them that are in here. If you haven't seen this article, it was in 2011, the British Journal of Pharmacology. It's a really great article where they're actually breaking down independently all of these different plant secondary metabolites that are in all of the different strains of cannabis and doing individual testing on each one of them. So indicators of poor plant health would basically be any insect damage you have, disease, rotting fruit, small fruit, deformation or stunted growth, too much moisture, low test weight, so you're not getting density of your flowers or density of what you're growing, early die down instead of dry down. Lots of times I see out in the fields, specifically with cannabis when I've been consulting on people's fields, where they'll instantly cut the nitrogen or they'll cut everything, and you'll see this quick dropping of everything where everything turns yellow. That's actually not what you're looking for. You're looking for die down instead of dry down, where everything just drops, because what happens is the plants will actually suck the lipids out of those leaves in the last couple weeks instead of this concept of flushing, and they'll actually, those are actual things that can be turned into plant secondary metabolites, but if they just go into die down, they're gone and they're not allowed in the reserve of the flow of the plant anymore. So critical points of influence. I'm not going to go through all of these slides because I know that there's not going to be enough time, but basically say for instance, any of your plants that you're growing, 9 to 12 days after emergence, you've basically, so when you've got a seedling that's that high, you've determined how much output it's going to grow no matter what you do to it. So after 42 to 49 days, there's really not much you can do to alter that. So if you're trying to, whatever type of plant that you're trying to grow, you can change things, but you can't critically impact that plant as if you were to give it the complete optimal growing. Plants have a signaling and behavioral, something that we had found this last year where we took several different species in one of my clients' fields, and we started them off in regular pots where they could go into being root bound, and then they were transferred into larger pots. The plants that we actually started immediately in 30-gallon smart pots, we were able to actually grow denser and fatter and more robust plants, because the plants actually had more ability to create more mass instead of ever going into a root-bound phase. So it's just giving people the awareness that even when they're in that seedling, if you get them into the right size pot and the right environment with the biology, you can actually affect what you're going to have in the end. So let's see here, this isn't going to correspond to this. So protein synthesis and proteolysis. The formation of complete proteins in a plant is the primary means of ensuring pest resistance, and basic mineral balancing and inoculation are key for setting up this level of functionality in our plants. Insects actually don't have the enzymes in their digestive tracts necessary for breaking down proteins and have antennae necessary to see if their preferred food crops are higher in nitrates and amino acids or proteins. Any problems that you see out in your field are usually excess salts or nitrates of some form that aren't broken down, and that's where your plant will actually start to show some sign of whatever negative things. Something that most people are completely unaware of is called paramagnetism or diamagnetism. Paramagnetism technically is the capacity of the charge in a material to align in the presence of an applied field. So green sand, rock phosphate, humates, basalts, pumice, any types of things that are from an organic mine source actually have a paramagnetic reading to them. They create like little magnets that go into your soil and create charges. Practically, plants are diamagnetic by nature, and so the stronger the paramagnetic charge of the soil, the stronger the effective opposite charge between soil and plant, which facilitates plant growth. So the more paramagnetic things that you work into your soil, they'll be there after you're gone, and they'll actually keep enhancing any plant life. ERGs is a way that you can test this type of a reading, and it's the energy released per gram second. It's effectively, this is the electrical energy flowing in the soil system that facilitates the mobility of the crop's nutrients and building of plants. One of the simplest, real-time, and least expensive ways to see the overall functionality of the soil with proper mineralization and the biological system support. This is a way to see if the battery is still being charged or running out of juice. There's a concept that you're in your soil that you've got a CD and you have a checking account. And so as you're working through your checking account of the minerals and the things that you've added for your quick response of that year, you also have things that are turning into your CD for the following year or years to come that take a longer time to break down. But it's really important to keep charging both of those. One of the things that we've saw in a lot of the crops that I was working on last year is that by just simply planting cover crops, even if it's in smart pots or wherever you're growing your crops outdoors, is to regenerate that soil by actually planting you can do crimson clover, white Dutch clover, hairy vetch, Austrian peas, fava beans. You can do like a nice mixture of these things. So right when you get your plants out of the field, immediately plant cover crops on there, and you'll be enhancing all of the nutrients that you put in there to be bioavailable in the springtime when you start back up. So sometimes people aren't really interested in getting into all of the bioinoculants for soil and mineral balancing. So this is just some basic things, and Ian's actually got a company called Biologic Systems that helps to guide clients towards simple methodologies to achieve things that will basically act as a chemical fertilizer but give you the same performance. So in like your drip system, you could be doing just a castings tea and enzymes, a sea mineral concentrate, some form of calcium and phosphorus and potassium, and then as a foliar, same thing, creating a compost tea and castings and a sea mineral concentrate and kelp. If you don't want to get into plant sap monitoring or soil conductivity testing, there's some basic recipes that you can do, and it's just like taking beneficial bacteria for our bodies where we take probiotics or enzymes, is that if you get in a rhythmical pattern of being able to apply these to the plants, you will actually see that all of the nutrients that you're adding to your plants are being digested in an effective manner. So where are we at on time?
Fine.
So we're going to go into you now. So one of the other things when he had asked about what is the biology in your garden, the biology really is a digestive tract of the plants. It's kind of, I look at it in the same concept of like fructooligosaccharides or acidophilus in your belly. And the symbiotic bacteria populations, there's all this nitrogen fixating happening. There's bacteria that are solubilizing nitrogen. There's phosphoryl solubilizing bacteria. There's all kinds of different zooplankton and algae. And then all of the different indigenous microorganisms. When we add chemical fertilizers to any of our soils, all of our indigenous microorganisms actually migrate away from that area. So the more that we start building the biology, we can actually see a translocation of the bacteria, where you'll actually see beneficial bacteria that were native in your soil that will actually come back to that area. Here's a simple local bioinoculant, indigenous microorganism, that I've shown a couple of farmers that they've had really good results. There's things that you can buy like EM and different things that you can do, but in a 5-gallon bucket, you can take and cover and fill it with tepid water and take different weeds or plants that are growing in the area like horsetail and comfrey, nettles, kelp, alfalfa, whatever you have, white oak bark, lichens, seaweed. There's all kinds of different plant life that you can do. You take this material and you can actually create like an extract from that and add that to your compost teas. A simple way to make enzymes or to create an inoculant you could take those same materials, that previous recipe where you're taking all of those organic materials. You take 1 cup of EM and 1 cup of molasses, and you cover this. It's an anaerobic environment for about 20 to 40 days, and the pH will drop between around 3.6 to 4. And that will stay good if you keep it in a cool, dark area for a long time. And then every time you're doing your compost teas, you can take a cup out of this, and it acts as like a kefir starter for creating yogurt. But every time you use this in your biological, whatever it is, whether it's compost teas or fertilizers or whatever you're using, it will help to activate them and create them more as a bioavailable form for the plants. So the reality is that regenerative farming, it's a long-term thing. It's not a quick fix. It works in harmony with nature to reestablish mineral balance and introduce and enhance beneficial microbiology in the soil. Regenerative farming uses conventional and organic techniques. It's not organic or not, and it allows, with a combination of chemistry, physics, and biology, to produce sustainable, nutritious crops. This is a slide that I got from Ian, actually. You want to explain this one?
Sure. This is looking at a rugby field in England. And you can see on the left the image on the top and the bottom. All right. There we go. All right. So we got a beat-up rugby field, and they started applying a compost tea on that field every month for a year, and you can see the field a year later on the right. And you can obviously see the difference in the color and a little bit less mud and a lot more grass. But I like to point out the roots. If you look at the top pictures, you see on the left side, the soil is really dense and anaerobic, and you don't see a whole lot of roots. And then when you look on the right, you can see how light and fluffy that soil is with all those bright white roots. So all that was done there was just adding in these beneficial organisms that are starting to work away at that soil and increase the quality of it. So.
Yeah, and one of the things is a lot of the growers that come over to see what I'm doing, everybody's always interested in some of these different projects where I'm consulting. They keep saying, why is it that your soil is so soft on top? We're talking the end of flowering. There's never any compaction. If you have ample biology in your soil, at the end of your grow, when you actually cut your plants down, your soil should be loose enough to where you can actually put your hands inside of it. It should never be packed down. And that's the same type of a thing here. It creates like a light fluffiness where it actually starts to create a biological soil crust. And then that's actually, you want to explain that one?
Yeah, you want to do this one?
Yeah.
This is a kind of cool slide if I can manage to interpret it for you guys. And what the theme is, before I even look at this, what the theme is here is that as you start to increase the amount of fungi that's in your soil, and that could be, thousands of different species of fungi, as you increase the fungal biomass, you can see a correspondence in the increase in the organic matter in the soil. So what this is looking at is data from the table grape growers in Australia, where what they did was they started adding a compost tea into their vineyards through their irrigation, with especially with a special kind of fungi in it, a mycorrhizal fungi. Kind of litmus test. How many of you guys have heard of mycorrhizal fungi? Anybody? Okay, well, it's a fungi that grows on the roots of the plant. It inoculates on the root of the plant. It actually forms its spores on the root of the plant, and then it grows its little roots out from the root of the plant. So this mycorrhizae colonizes the roots, grows out from the roots, and is able to go out and mine minerals in the soil and bring them back to the plant. And it also works to defend the plant from pathogens. Well, what that mycorrhizae eats is it actually eats foods that the plant feeds it. So the plant is sucking all this carbon out of the air, right? CO2, sucking it into the plant. Well, a third of the CO2 that the plant is taking out of the air, if you have this mycorrhizae growing on the roots of the plant, is pumped into the soil and is fed to the mycorrhizae. So, you know, a third of that CO2 the plant's sucking out of the air is being fed to this organism, right, in the form of proteins or complex carbohydrates that the mycorrhizae is eating, and the mycorrhizae is in turn going out and finding calcium and zinc and boron and selenium, all these things, bringing them back to the plant, and they have an exchange. So we describe it as like it's like a microbial marketplace that's going on, where the plant is exchanging these, they're called exudates, these foods that it's manufacturing, to these organisms that are going and finding all this stuff for it in the soil. So what's going on here is they've inoculated the soil, they've added this biology in there, and that plant is pumping carbon into the soil, which is increasing the carbon levels. So you see the carbon's going from 2% up to 6%, right, over 3 years. If you were to try to do that by adding compost, that's the main way that people try and increase the organic matter in their soil. These guys would have had to be putting out probably millions of dollars worth of compost per acre to try and get the organic matter to go from 2% to 6%. But they were able to do it just by getting these organisms in the soil that are utilizing the carbon from the plant, that the plant is sucking up from the atmosphere, in order to increase the carbon level in the soil. So that really has pretty dramatic implications in terms of where we're headed with agriculture, because the whole problem right now is we're losing all the organic matter in our soils through tilling and the use of chemicals and the compaction and ag. We're flushing all the carbon back down into the ocean, and our, you know, the NRCS says we have like 30 years left until we've eroded all of the topsoil off of our continent. Well, what really makes the component, what makes topsoil, is the carbon that's contained in it. So we have a mechanism here by adding this biology back into our soil to jumpstart this process where we can start increasing the carbon sequestration in the soil. So you look at the big picture right now in our situation as human beings on this planet, where we have this legacy load of carbon that's in the atmosphere, all right, and we're depleting our soil so rapidly that potentially in 30 years we won't have any food left. And really the only solution I see, you know, to both of those problems, to the food crisis and the legacy load of carbon in the atmosphere, is to get all that carbon back in the soil, right? And how are we going to do that? It's by jump-starting this biological process of getting these organisms to work with the plant to start building carbon in the soil again. So long story short here, as you build the fungal biomass in the soil, as the fungi increases, right, the red, right, we're getting larger fungal biomass, you can see a correspondence with the increasing of the organic matter in the soil. So does that make sense, you guys? Is that somewhat? Okay. All right.
Yeah, so this is my last slide. I mean, just to kind of give you an idea. That's the hand.
Get close up there.
That's the hand that's on the base of that plant. So that's done with biology. There's no chemical fertilizer there. Plant yielded between 8 and 9 pounds. 48 of them in one field, all with biology. And the soil was as loose as could be. So how do I flip it to yours? So the biological soil solutions that are in stores, Ian and myself aren't saying that any one of these are better than the other, but we know that they work. There's a lot of stuff that's sold in the industry. I call it puff powder. I've got a microscope at home and even an electron microscope in a lab that I work at, and most of it has nothing in it. They also have things that are host specific. So like there's specific things that you'll see on some of these products where they show all these strains to make it look very complicated and sophisticated. One of them is called rhizopogon. Rhizopogon is actually a puffball mushroom. It's host specific for a pine tree, but it's sold in 7 different products that are on the shelf for medical cannabis in the hydroponic stores. Progress Earth, Organic Pacific Grow, Beneficial Biologics, RTI's products are absolutely off the hook. I highly suggest you implementing those. They're Azospirillum, they're Mycos, all of their different products. Bounties are pretty good. I haven't actually worked with them. I've just seen some things about them. Acadian Seed Products. Biologic actually carries California Humus and several different unbelievable products that are quite good. And then the bottom one is about a Vortex brewer. That's on a whole nother level, but I'm going to flip it into Ian's slides right now. How do you flip this?
He was asking about soils.
Coming soon.
You can just flip over to the.
This one
Should be on there somewhere.
Yep.
Yeah.
Here.
Here, come on up.
Someone want to pass this?
The field broke down and the tea that you used cleared it up, the field that you were
So he was asking about the that plant we were looking at. Let's see if I can phrase that right. If the biology in the soil was able to help deal with the compaction, and what was the condition of the soil before you started?
It was basically a lawn, acre-and-a-half lawn behind someone's house, where we went in and we basically took a rototiller. We added compost to the top of it after we cut down or scythed the top, allowed everything to turn yellow, sprayed it with compost tea, added compost and earthworm castings and some pumice because it was in the region of Oregon, tilled that in, came back and spaded it. A spader actually acts like a bunch of shovels, where a rototiller implodes the soil. A spader actually loosens up the soil and acts like a bunch of shovels going in. Then all there was just some wooden frames built up on the side, and the plant was planted in that. So at the end of the year, there was really no soil compaction, and all of those rows had cover crops. So all it was grass for however many years that it was someone's backyard lawn. So it was its first year. So.
So we've got 15 minutes, and I think I'd rather take your questions, really, rather than looking at the slides here, because we've covered most of this information, and I'm guessing that you guys, your questions will probably fuel the conversation a little bit better than me going through these slides.
Does anybody want to have him explain about the biology in some of these slides?
Well, I can do this, but
yeah,
again,
that's why I'm asking.
Yeah. You're making it.
Do you guys want to know a little bit more about what's going on with the protozoa and so forth in the soil? Yes? Yes? Okay.
Okay. All right, I'm going to try and give you guys some information that will make sense here. And I do want to get to their questions, so I'm going to just go through this really quickly. Yeah. So Christopher kind of blew over this slide earlier, and to me it's probably the most important one out of all of them you'd see today, and it's just really understanding what the basic functions that these microbes do in the soil, what they can do for you. And so I'll just talk about each of these 7 here really simply. So the organisms in the soil are able to maintain the aerobic conditions, and why that's important is that soil, in order for it to function properly, it needs to be an aerobic environment, meaning you need to have lots of oxygen, and that's really where the whole hydroponic study began, was once they realized, wow, the more air you get into the root system, the healthier the plant can grow. In a soil-based system, all these organisms, when they're growing, they make a glue. They make a slime layer all over themselves, and it's really that glue that's what holds all the particles in the soil apart and keeps the space in between them. So when Christopher was talking about that plant that was growing, you know, in that soil that's really light and fluffy and aerated, you know, what's keeping that soil aerobic is all these organisms that are growing in the soil and the glues that they make. And you'll have, like, for example, as an earthworm goes tunneling through that soil, it's making a passageway where air is able to get down into the soil. As the fungi is growing its hypha, which is like the roots of the fungi, all through the soil, these strands, again, they're making passageways where the air is able to get into the soil. So, you know, we've seen soil-based systems that have just as good of the ability to infiltrate oxygen, just like a hydroponic system would. You know, where you would think that the soil would have a heaviness to it. Well, not necessarily. If you have the biology in there, it keeps the soil aerobic. Anaerobic conditions, if you get a compacted condition in your soil, let's say in an outdoor garden, you start getting kind of a crust on the top of the soil from putting down fertilizers and whatever. If you reduce that oxygen, the bad organisms start to grow. So, you know, you can there's a dividing line in the world of all these organisms of ones that grow in an oxygen-rich environment, an aerobic environment, and ones that grow in an anaerobic environment. And basically all of the pathogens are anaerobes. So if you maintain an aerobic condition in your soil, you're not going to have problems with fungal diseases. You're not going to have root-feeding nematodes, because those organisms really thrive in an anaerobic condition, right? So very important. And then the second function is they actually build the structure of the soil. So not only can they maintain it, right, but let's say we've got a farm field where the soil's all been devastated, it's all compacted and anaerobic. We can put these organisms back in that system, and they right away start to work to aggregate that soil again. There's certain sets of organisms that can go into those aerobic conditions and really start to turn it around. So, you know, not only can they maintain that structure, but they can build it where it's been lost. And the third function is suppressing disease. So, let's say, you know, my hand here is the leaf of a plant, and that plant is pumping out these exudates, like we were talking about a moment ago with the mycorrhizae. It's pumping out all these sugars and carbohydrates and proteins onto its leaf surface to attract these organisms to come in and colonize it. So if you look at a healthy leaf under a microscope, you actually can't even see the surface of the leaf. You'll see a layer of, you know, 10 organisms thick, these bacteria and fungi all piled on top of each other, protecting that leaf. So now let's say you got a little powdery mildew spore that comes and lands on that leaf surface. You know, is it going to have any chance of getting colonized on that leaf? No way. It's going to get gobbled up and kicked out of there long before it can do any damage. Now let's say we got this leaf surface where we've come through and we've sprayed it with some type of fungicide or pesticide. Well, let's say we're spraying with a pesticide, right? Is that just going to kill those little spider mites? No, it's not. It's going to kill all these beneficial organisms that are growing on that leaf surface too, right? So all of that biology is now dead, and now you got a little mildew spore that comes and lands on that leaf. Well, it's going to make itself right at home because there's no biology there to defend the plant. So it's really important to understand that plants have an immune system just like you do. You know, the whole surface of your body is covered with biology. If you're to look at your skin under the microscope, you're going to see a layer of organisms there that are working to protect your skin, working to do things like breaking down your body odor, etc. You're covered in this biology too. Even by count, 70% of all the cells in your body are actually bacteria, right? By count, not by mass, but they're so tiny, right? So you're technically more bacteria than you are even human, right? So next time you make a mistake, you can just blame it on your bacterial biomass, right? So, you know, plants have an immune system, and once you compromise that immune system, whether it's using pesticides or even in the soil, using too much nutrients, the plant has a compromised immune system, and now these pathogens come right on in. So that's one of the big untold stories of fertilizers. You know, well, you use a lot of fertilizer, the plant grows really fast, bright green, but now you got mildew. Now you got botrytis and spider mites and all these pathogens that are taking advantage of that compromised immune system. So, you know, you got to really watch out with the fertilizer use because too much of it is definitely can be a bad thing. you start compromising the immune system of the plant. So we really want to find that balance point of utilizing fertilizers, but really relying on these microorganisms to do most of the work, so that your fertilizer is just kind of an addition. It's like a little rocket fuel that you're adding into the system. So fourth function is decomposition. Right? If microorganisms didn't break down stuff, the planet would just be covered with a layer of dead things, probably stories tall. Right? We need these organisms so that every example from cutting the grass in your lawn, you need that biology that's going to break that stuff down, or after years you would just have a layer of dead stuff there. So a pretty important function there. You think about in your gardens, at the end of the season, when you rip that root ball out of the ground, if you got good biology in the soil, all these organisms, right, they're going to start digesting all the root material you leave in the soil, and you're actually setting yourself up for the next year where all that good biology that was growing all over your roots is left there and is left behind in the soil. So myself, I don't actually remove the roots. I leave them there, and I'll keep the soil moist over the winter, and that'll break down all those roots. Then the next year the soil's even better than it was before. So if you're doing this properly, the days of replacing soil are over. Your soil should get better every year. You don't see farmers out in a farm field going out there and loading up their soil into a truck every year and dumping it, taking to the dump, right? It's pretty much only in the indoor gardening world where you actually see a cycle of people replacing their soil. And why do they have to do that? Because they're loading it with all these soluble nutrients, these fertilizers that are building up the salt levels in the soil, and then you have to replace it because it becomes toxic to the plant when you get all those salts. Do you have a question? He's saying, for that reason, do you recommend not turning the soil? What I recommend is don't turn it until you're going to plant in it. So over the winter, I'll just leave the soil, and I'll usually put a layer of alfalfa on top of it, nice thick layer, maybe 3 or 4 inches thick. And alfalfa is like caviar, caviar for earthworms. So they'll come up, they'll start grinding away at all those dead roots and eating up the alfalfa, and then by the springtime, you'll have a layer of worm castings on top of your soil, maybe like a 1/2 inch thick. So, you can utilize that overwintering of your soil as really an opportunity to increase the quality of it. And that's, rather than cover cropping, I generally use that alfalfa mulch. Because in my soil, I found if I try and cover crop it, I come back the next year and the roots have grown so much. If we try and do clover, I'll have a big solid chunk of clover roots. So I prefer to use that mulching and litter layer method. So did you have a
Yeah, I was just going to share that I saw a couple methodologies where basically people just grew hairy vetch inside their field. There's some crops like white touch clover and grasses that are very invasive, that once they're there structurally, it takes you turning them over. They actually produce exudates that are destructive to the types of annual crops that we're producing. But if you take hairy vetch, for instance, and fava beans, in the springtime you can go out there before they start going into flowering, because once these cover crops go into flowering, all the nitrogen that they've fixated in the soil is actually partially leaving and going up into their flowerets. And once that's been pollinated, some of that nitrogen, all these little pods of nitrogen that are nitrogen fixated in the soil, will actually turn into crops for the following year. But what you can do is you can go out, scythe that area. So say you've got an area this wide where your plant was growing last year, say you've got a 10-foot plant, wherever your plants are, however your spacing is, you can go in and you can actually scythe that area, sheet mulch it or mulch that area, and just dig a hole where your plant's going to go. There's no reason to disturb and turn all that soil. So
yeah. So the last couple of functions here. Nutrient immobilization, that's a fancy way of saying that these microbes are really good at keeping all the nutrients in the soil and keeping them from leaching away. So let's say you don't have good biology in your soil and you go and you apply some calcium, right? Well, the majority of that is just going to wash away. When you go to water the plant, that calcium is going to be soluble, it's going to wash away. Well, if you have all these critters in the soil, a healthy population of them, they will hold on to that excess. So you can put some calcium out there. Whatever the plant doesn't take up, the fungi that's in the soil will grab onto it and hold it and keep it there. And will most likely eventually do what with it? Well, it'll take it to that microbial marketplace and exchange it. It'll exchange it with the plant for a little bit of the exudate that plant creates. So immobilization, that's a big part of the efficiency that we're seeing in these systems, where you can use a very small amount of fertilizer because it's staying in the system, it's not washing away. And then mineralization is then the process of making those nutrients available. So like there's a really neat example in terms of these two organisms in the soil, bacteria and protozoa. I mentioned this briefly a minute ago, that in a healthy soil, in a gram of a healthy soil, you might have 1 billion bacteria and 200,000 of these little protozoa, right? Those protozoa eat bacteria all day long. That's all they eat. So one protozoa will eat 10,000 bacteria in a day, right? And you have 200,000 in a gram. So that's like 500 million bacteria or something like that are being consumed every day in 1 gram of your soil by those protozoa. Oh, what the heck does that mean? Well, those protozoa, as they chomp away at all those bacteria, they're pooping out the nitrogen that's contained in the bacteria in a soluble form that plants can uptake as NH4, ammonium, right? So this mineralization process is basically a way that free fertilizer can be generated in your soil. So nitrogen, for example, in my gardening, I don't use any nitrogen fertilizer at this point, because in my soil, I've now got like 500,000 of these protozoa per gram that are producing like 10 times the amount of nitrogen my plant can even uptake, right? And what happens with all that excess nitrogen? It just gets sucked up by the biology again. The bacteria scoop it back up and hold on to it. So you're creating this cycle where the nutrients are being released. Whatever the plant doesn't take up, the biology is holding on to again, as opposed to a chemical system, right, where you don't have the biology in place. Let's say a soilless system or an ag field where the biology's been lost. You're adding that fertilizer onto that field or into that system. Well, if you don't have biology there to hold on to it, the majority of it's just going to wash away. So I'm getting off track, but mineralization is the ability of the biology to free up fertilizer for your plant's uptake. And the 7th one, fixation, Christopher was kind of alluding to that one for a minute. Nutrient fixation mostly applies to nitrogen, and what we're talking about is the ability of these organisms to suck nitrogen out of the atmosphere and ultimately make it available to the plant. So a good example of this is a product that we sell that's called azospirillum, which is a type of bacteria that can fix nitrogen out of the atmosphere and make it available to your plant, right? Now that's pretty profound implications there, and that organism can repopulate and grow in your soil. So you can add, when you're planting your seed, you put 1 gram of that bacteria inoculant in your hole, and that stuff immediately starts growing and multiplying and sucking all this nitrogen out of the atmosphere and feeding it to your plant, which is a pretty incredible equation in comparison to buying a bottle of expensive nitrogen fertilizer and having to pay to do that process. So, and this stuff is, this isn't your grandma's organic here. Like, there's a huge revolution happening right now in the understanding of how all this biology relates to the way plants grow. And it wasn't 15 years ago that there was barely any awareness at all these beneficial organisms. Now we know that 99% of all the organisms that grow in the soil are good guys, are beneficial. Whereas, it was only 20 years ago that they figured 99% of them were pathogens, right? So the whole science is this evolving world where, we're just now really being able to see in there and really see what's going on and realize, holy cow, we really missed the boat here. We thought all these organisms were problematic when really they're beneficial. So I think we're pretty much out of time. Was she saying we got to skedaddle there?
Yeah, she is. And one thing I will suggest before we finish up is that over at our booth, if you want to come over and look through the microscope and see some different things about some biological products that are on the shelves or some roots or anything, learn a little bit more, listen to an actual machine that we have over there that's playing the sound of cannabis sativa, the actual sound that the plant is making. We have a machine over there. You can hear the music of it, a new spinning light. and just learn a little bit more about the offerings that Biologic Systems offers and Soils offers. And so, is there any specific questions as we're finishing up?
Websites. Yours is
soils.com, but again, that's coming soon. BiologicSystemsUSA.com and reforest.com.
I'd say come catch us over at the table, and we can try and make a little more sense out of all this for you guys. So. I think I got your question. He was asking about, we were saying that the chemical fertilizers can kill the beneficial organisms, and it's all about threshold. So we work with people all the time that utilize synthetic nutrients. There's a certain threshold, though. You can use those products at a low dosage, but once you start getting really high levels of salts, meaning soluble nutrients in the soil, and it's there's no fine line, there's no magic level, but typically what we do when we work with people using synthetics is we say, okay, cut back what you're using in half, and then start utilizing these beneficial organisms, and you'll generally see the same result. You're going to make way better use. So whether you're using an organic fertilizer or a synthetic, doesn't matter at all. If you have a high phos product that's a 0,13,0 bat guano next to a high phosphorus synthetic fertilizer that's a 0,13,0, you have the same amount of chemically available phosphorus. It's the same stuff, right? So you can have the same problem. Like if you're using an organic fertilizer, you can use so much that it will have a detrimental effect on the organism. So, typically what we promote is we just call it the cutback method. It's, cut back what you're doing right now. Cut back the amount of fertilizers, the amount of pesticides, reduce that stuff in half. Add the good stuff in, and and potentially you can see that same result and have it be a lot more cost effective. So we should wrap it up here. We're over our time, but come hang out at our booth and talk more about this. So.